In this video, we will discuss the tutorial questions from the workbook Biology. We will focus on question number 1, number 3, and number 4. Now, let's begin with question number 1. This diagram is related to the step or method in gene cloning. It consists of isolation, cleave, insertion, transformation, and amplification and screening process. Remember the acronym for the methods in gene cloning, ICITAS. Now let's take a look into the question. We have the enzyme P, we have the molecule R, we have the process Q, process S, and process T. Let's answer the question. Number 1A. Name enzyme P and the enzyme involved in the production of molecule R. Both target gene and plasmid appear to have a blunt end, so the restriction enzyme used for this process is SMA1. Target gene and plasmid seal by DNA ligase to form molecule R. The next question. Name molecule R. Molecule R is recombinant DNA or recombinant plasmid. Process Q is insertion because the target gene inserted into the plasmid. Process S is the transformation because recombinant plasmid reintroduced into the bacteria. Process T is the amplification process because this process produces many clones of bacteria. Question D. The next step after the amplification is screening. This process will detect bacteria that carry recombinant plasmid. The next question, state 3 benefits of insulin produced by genetic engineering. The target gene is from human, so the insulin produced is similar to human's insulin. So it is non-allergic or has no side effects. It's cheaper or cost efficient. It can be produced in large amount, and it is halal from the Muslim perspective. The conventional way to produce insulin is to extract insulin from the pancreas of slaughtered animals such as cows and pigs. Here is the suggested answer for this question for your own reference. Let's continue with question number 3. The diagram shows the process of the formation of cDNA from the mRNA. So the cDNA synthesis can be represented by the following diagram. This diagram shows the cDNA synthesis from the mRNA. Let's focus on the questions. We have the mRNA, and then we have the ssDNA plus mRNA. We have the degraded mRNA, and then we have the cDNA synthesis that produce many copies of cDNA. Now let's answer this question. 3A. Identify A. A is mRNA, consists of exon, that is coding sequences. And then B, the enzyme Y is reverse transcriptase to synthesize cDNA from mRNA. C is a double-stranded DNA. The DNA polymerase synthesizes DNA strand from the single-stranded DNA. Question C. The application of cDNA. The cDNA can be used as a target gene and useful for cDNA library collection. Process D is amplification. This process will produce multiple copies of double-stranded DNA through polymerase chain reaction. The polymerase chain reaction will require target DNA from the double-stranded DNA, pre-nucleotides, primer, and take polymerase. The characteristic of the take polymerase that is used in polymerase chain reaction is it must be thermostable or heat-resistant DNA polymerase. Here is the suggested answer for question number 3. Now we will answer question number 4. From the diagram, it shows the step in the gene cloning that will involve plasmid as a cloning vector. The step in the gene cloning will involve isolation, clip, insertion, transformation, amplification, and screening. From the diagram that shows the plasmid as a cloning vector, it will involve the step P, step Q, and then the step R. Now, let's answer the question. Question number 4a. Identify step P, Q, and R. Step P is insertion, in which target gene is inserted into the plasmid, while step Q is transformation, in which recombinant plasmid reintroduced into the bacteria. Step R is the amplification and screening. This process will identify and multiply the bacteria with target gene to produce functional protein. For question number 4b, the process is insertion. During this process, both target gene and plasmid cut with the same restriction enzyme to produce compatible or complementary ends. After that, the DNA ligase is used to join the phosphodiester bond between the target gene and plasmid. The characteristic of plasmid as a cloning vector. First, it has multiple cloning sites, or MCS, able to replicate freely because of the origin of replication gene, and possess selectable genetic marker, ampicillin-resistant gene. For the question number 4c, the characteristic of recombinant bacteria. Recombinant bacteria is the bacteria that already received recombinant plasmid. It's become resistant to antibiotic due to the presence of plasmid. It's able to express the target gene. It's able to maintain the structure of recombinant DNA and able to amplify the gene of interest. Do not confuse with the characteristic of the recombinant bacteria with the characteristic of the bacteria as a host cell. When the bacteria receive recombinant plasmid, now it's become recombinant bacteria. Please refer to this following suggested answer for your reference.